Hi guys, this is Jane. Today I'd like to share on the Western honeybees. You probably learned a lot about them before, but I promise you that you're gonna find out more that you don't already know. So let's. The Western honeybee, also known as Apis mellifera, it falls under the class of Insecta under the family of Epidae. They are one of the earliest insects domesticated by humans at about 9,000 years ago, particularly the Egyptian and the Greeks. Besides, they are also one of the 5 species in the world that is not replaceable. So what do these honeybees feed on? They feed on nectar, pollen, honey, and propolis, and they also help to pollinate 85% of our plants. While the Apis mellifera is not categorized as endangered under the IUCN Red List, like some of their cousin species, they do portray some significant genetic flexibility that we were going to be talking about, especially when it comes to neurogenetic, where they are age polytheism, which means that they behave differently at different age, and surprisingly political, given they are just little insects. As most of you know that bees, especially the honeybees, they live in colonies, and the one colony is made out of a queen, usually, and drones and worker bees. So how big is a colony exactly? Usually, it depends how big a hive is. A colony should make up to about 20,000 to 80,000 bees. The number of bees also depends on the season. In warmer days like spring and summer, there'll be more bees in the hive, whereas winter days, there'll be a lot less. Before we dive into the essence of this presentation, let's take a peek at the life cycle of a bee, shall we? Here is a bee egg as it hatches into a larva. Those newly hatched larvae swim around their cells feeding on liquid food that the nurse bees secrete for them. Then, their head and their legs start to differentiate as they transform into pupae. And here is the same pupation process seen from above with pheromones running around the cells. Next, the tissue reorganizes in their body and the pigments slowly develop in their eyes. In the last step, the skin shrivel up and they start to sprout hair over their body. In a honeybee colony, there are 3 types of bees. There are the male drones, but in this video we're not going to talk much about it. And then there are the female bees. Most of these bees are worker bees, but a small number of the female bees will become the queen, and they'll go on and find new hives as they mature. So now, most of these female bees have the same mother, and some even share the same fathers. So therefore, genetically, they are very closely related to each other. But how can essentially genetically identical bees be so different? The worker bees live for between 22 and 42 days, though in some cases they do live a little bit longer. On the other hand, the queens can live up to 2 and 7 years. Other than that, the queen bees are also much larger than a worker bee. A full-grown queen is about 2 centimeters, whereas a worker bee is only half the size of a queen bee at 1 centimeter long. And another major difference between the queen bee and the worker bee is queen bees have fully developed ovaries that they will use to produce eggs to provide the next generations of honeybees. The worker bees, on the other hand, have very underdeveloped ovaries and are in fact sterile. The main difference between workers and queens, however, is their role within the hive. The workers made up the majority of the bees in the hive. They don't breed, and their role is to look after the hive, including the larvae, and going around foraging from plants in order to feed the hive and the queen. While the queen doesn't move from the hive, and her main job is literally to just produce eggs that will go on to become the workers and the future queens. Here comes the big question. So what makes a queen? As we know that both the queen and the worker bees come from the same colony, which means that they are genetically identical since they share the same mother, and some probably share the same fathers as well. So the question is, who gets to decide which larva turns into a queen and which ones turn into the worker bees? Does the genetic decide what we become? In this case, it doesn't quite make sense, does it? Thanks to all the brilliant scientists around the world, they found the answer, which I'm going to share with you in just a little bit. The answer is epigenetics. In order for us to understand the concept of epigenetics, in this video, I'm going to try my best to introduce the concept of epigenetics and how it works with bees. So in order to explain this, we need to understand the role of DNA. In short, the DNA in our cells is a set of instructions. It tells the cell how to make protein, and proteins do pretty much everything in our cells and our bodies for that matter. A section of DNA that codes for a single protein is called a gene. In this case, to not sidetrack, we just have to know that epigenetics plays the role in switching on and off specific genes. In relation to bees epidemic, we need to know that switching genes off happens by a process called DNA methylation. On the other hand, genes are switched on by a process called histone acetylation. So switching on and off genes make sure that cells only have the genes that they will use switch on through their life. So what caused this huge difference in the lives of different bees that are incredibly closely related to each other? We've known for some time that honeybee larvae fed on royal jelly become the queens. Well, that's mostly the case. All bee larvae are fed on royal jelly for the first couple of days, and then the bees that will become workers are then switched to a diet of something called bee bread, which is a mixture of pollen, honey, and nectar. The larvae destined to become queens will remain on a diet of royal jelly for the rest of their lives. Well, in one study, researchers added a chemical to bee larvae. This chemical is called small interfering RNA, and this is a chemical that switches off or silences specific genes. And the specific gene that this interfering RNA silences was the DNA methyltransferase gene. If you remember, this gene normally codes for the DNA methyltransferase enzyme, which adds methyl groups to the DNA, effectively switching off certain genes. With the DNA methyltransferase gene switched off by the interfering RNA, the researchers found that a higher proportion of the larvae developed into queens. In the larvae which had the DMNT gene switched off, most of them turned into queens. Where the DMNT gene was left on, the larvae turned into workers. Besides, the researchers also found that the pattern of the genes that were switched on mirrored the pattern of genes switched on when the larvae were fed royal jelly. This is suggested that the royal jelly had some kind of epigenetic effect on the larvae, turning them into queens. Well, that's not the end of the story. A further experiment did not find that royal jelly had anything to do with the larvae turning them into queens, but they, however, did find a fatty acid called 10-hydroxy-2-deconoic acid. In short, we're just going to call it 10-HDA. They found that 10-HDA doesn't stop DNA methylation, but instead it influences histone acetylation. If you can recall earlier in the video, I mentioned that this process is when the genes are switched on instead of off in the cells for certain functions. In this case, 10-HDA has been found to inhibit the deacetylase enzymes, stopping them from working. That means that if they can't remove the acetyl group from the histones, the DNA is wrapped more loosely, making it easier to get the DNA and to read it, to make protein. It appears that 10-HDA is the active ingredient in royal jelly, stimulating the larvae to turn them into queens. And there's another interesting findings about pollen and honey. Scientists found that pollen and honey contain a substance called p-cumaric acid, and this substance is absent from the royal jelly. In a further experiment, bee larvae were fed with royal jelly, to which had been added p-cumaric acid, and they found that the adults that developed those larvae had underdeveloped ovaries. So it appears that maybe royal jelly not only nourishes the queen larvae, but protects them from the effects of p-cumaric acid that the worker bees larvae feed on. In another more interesting finding, bee bread actually has something to do with it. Bee bread contains pollen, and pollen is the plant's male sex cells, and these are used in plant reproduction, meaning that pollen contains the plant's genetic material or DNA. It also contains pieces of genetic material called microRNAs. These are small fragments of RNA, and it influences the expression of plant genes. It also affects the genes expression of bees that eat the pollen. To test this, researchers made some synthetic bee bread that didn't contain any microRNAs. To some of the bee bread, they added microRNAs from flowering plants. They then feed the bee larvae on the bee bread. So the bee larvae that were fed with the bee bread containing microRNAs were smaller in size and had underdeveloped ovaries and were more like worker bees. Of course, they also fed some of the larvae with the bee bread that doesn't contain any microRNAs, and the bees grew up to be larger and with more developed ovaries. In another word, more like queens. So this suggests that the microRNAs are influencing the epigenetics of the bee larvae. The actual answer is probably a mixture of all the effects that we have just discussed today, and possibly some of those that we haven't yet learned in biology. So that's it for today. I hope that you have enjoyed the video. I've included some of the references that I studied for the making of this video. I hope you'll find this useful. Thank you. Bye.